Welcome to Botany class, Chapter 1, Transport in Plants, Part 5, Transport of Mineral Nutrients. In this, carbon and most of their oxygen form CO2 in the atmosphere. Nutritional requirements are obtained from minerals and water in the soil. All minerals cannot be passively absorbed by the roots. Two factors account for this. One is minerals are present in the soil as a charged particles, ions. Second is concentration of minerals in the soil is usually lower than the concentration of minerals in the root. Here, ions are absorbed from the soil by the passive and active transport. Specific proteins in the membranes of root air cells actively pump ions from the soil into the cytoplasms of epidermal cells. Endodermal cells have many transport proteins embedded in their plasma membrane. Ions have reached xylem through active or passive uptake or a combination of two. Their further transport of the stem to all parts of the plant is through the transpiration stream. Here, the chief sinks for the mineral elements are the growing region of the plant, such as apical and lateral meristems, young leaves, developing flowers, fruits and seeds, and the storage organs. Unloading of mineral ions occur at the fine vein endings through diffusion and active uptake by these cells. Similarly, before the leaf fall in deciduous plants, minerals are moved to other parts. Elements mostly readily mobilized are phosphorus, nitrogen, and potassium. Here, xylem exudates shows that through some of the nitrogen travels as organic or inorganic ions, much of it is carried in the organic form of as amino acids and related compounds. Phloem transport. Flow from source to sink. Here, so sucrose is transported by the vascular tissue phloem from a source to a sink. The source is understood to be that part of the plant which synthesizes the food. Leaf, sink. The part that needs or stores the food. Here, the sink may be bidirectional or unidirectional movement from the phloem or a xylem. So here the phloem is upwards or downwards bidirectional and the xylem is only unidirectional, upwards only. Here the experiment which is shows illustration of mass flow by Munch. The pressure flow of mass flow hypothesis proven that sink is called as a mechanism used for the translocation of sugars from source to sink is called as the pressure flow hypothesis. Glucose is prepared at the source by photosynthesis, converted into sucrose. The sugar is then moved in the form of sucrose into the companion cells and then into the living phloem sieve tube cells by active transport. This process of loading at the sucrose source produces a hypertonic condition in the phloem. Water in the adjacent xylem moves into the phloem by osmosis. As osmotic pressure builds up, the phloem sap moves to area of lower pressure. The sink osmotic pressure must be reduced. Again, active transport is necessary to move the source out of the phloem sap and into the cells which will use the sugar converting into energy, such as starch and cellulose. Here, phloem tissue is composed of sieve tube cells which form long columns with holes in their end walls called sieve plates. Cytoplasmic strands pass through the holes in the sieve plates, forming continuous filaments. As hydrostatic pressure in the phloem sieve tubes increases, pressure flow begins and the sap moves through the phloem. Meanwhile, at the sink, incoming sugars are actively transported out of the phloem and removed as a complex carbohydrates. The loss of solute produces high water potential in the phloem and water pass out, returning eventually to the xylem. This simple experiment called girdling is conducted to identify the tissue through which food is transported. Here, the simple experiment shows that phloem is the tissue responsible for translocation of food, and that transport takes place in one direction towards the roots. This experiment can be performed by ours easily.